Good morning, everybody. We're going to go ahead and start this morning's session. And you're going to be hearing from a number of experts with diverse opinions and thoughts about combination therapies in particular and how that might look in Duchenne muscular dystrophy. And I was going to just start this session, and I'll let each person introduce themselves first, and then I'll show you a little bit of a slide presentation, and then we'll open it up to questions and answers and thoughts from the. I think your mic is live. So why don't we start on this end? I'll start. I'm Stan Nelson. I'm co-director of the Center for Duchenne Muscular Dystrophy. I also have a son with Duchenne who's 14 years old. Combination therapies are an intrinsic part of what we do in medicine, but they're not often fully explored in reality in the full clinical trials that we use to guide our hands. So actually, let me start at this end. And Lee, so we'll let everybody know who's up here.
Lee Sweeney, I guess from the University of Florida now.
I think you have to say that.
Hi, Joanne Donovan from Catabasis.
I'm Craig McDonald from the University of California, Davis.
I'm Nagaraju from the Children's National Medical Center here in Washington, D.C.
John Porter, Parent Project Muscular Dystrophy.
Cliff Bechtold, Bristol-Myers Squibb, genetically defined disease area.
Edward Kaye, Sarepta Therapeutics.
Thank you, guys. So you can see that's a diverse group of people who will have, I believe, rather different opinions along the way in this process. So one of the things that I thought this slide was very telling from, I don't know if this is going to work. All right, perfect. This is a slide from Parent Project Muscular Dystrophy of a range of different compounds that are in clinical trials or strategies that are in clinical trials, largely as monotherapies. And so in this list is a number of different drugs. And what will this look like as these are hopefully getting approved and actually used in clinical practice? How would we be combining them? Will we be combining them? There's almost no question we'll be combining them. And how will that look? What makes some of this exciting is that in other fields like HIV or AIDS, there's already been great success. This is there we go. This is when monotherapy for anti-AIDS medicines, HIV, viral suppression therapies are occurring, and this is when combination therapies first start being used, and during this whole phase, various combination therapies are being used. And this is the number of AIDS deaths occurring during this range, and they absolutely decrease the viral load, and that absolutely correlates with patient survival. Another area, and I can't read this slide because I took my glasses off, but this is in my prior field of childhood cancer. So in childhood cancer, we often bring out the success of childhood leukemia treatment, which is absolutely true. Most kids diagnosed with what used to be 100% lethal disease, and 100% lethal disease very quickly now survive, and they are cured. They live a very long, healthy life, and the field itself works on trying to minimize the long-term consequences of the chemotherapy that's given over the course of many years. However, I would caution you to be thinking about the fact that the first paper in this realm really establishing this field and the thought was published in 1948. And throughout the 50s, every single patient with childhood leukemia passed away from it. And it wasn't until the 60s and 70s that combinations and a systematic search through that combination space of a set of 6 different agents that were clinically effective on their own got worked out until reasonably large fractions of the kids were effectively treated. I'd say, and by the time from 1948 to say 1998, a 50-year period, there was a great success. That's great. 50 years is not great. So I think one of the things we should be looking at here is the logic and the thought process of how do we expedite that? What have we learned over those 50 years? What are we doing now that could make that process much more effective? So I'd like you guys to challenge the committee here for their thoughts on that. So oftentimes, how we do combination therapies in practice is This is a little diagram here where you might have drug 1, drug 2, drug 3, and each drug is set to its maximum tolerated dose, and one would back off from that somewhat to have an effective dose. And then we'd look at the combinations of those at their highest dose. Sometimes that's not the right place to look, and we might look at that in animal models before moving on to clinical trials, and then in small and large-scale clinical trials. But if we've missed the right combinations, we wouldn't be getting to the most effective agents. There are mathematical modeling strategies. There's ways to sample this very large space. I'll point out, actually, I may skip by this, but can you find the right combinations amongst a host of individually active agents or agents that you wouldn't know are active by searching more blindly than we would typically do? And I think there's some promise for that. So we can come back to some of these slides. But this is a mathematical model developed by a group of engineers at UCLA that are using a sparse data set. So instead of the millions of possible different combinations, they might sample just dozens to infer what the right combination is of a multi-drug regimen, is the example that's being shown here. The other way that we can learn is simply by looking back at data. And we do this all the time in clinical medicine. So this is an example of Duchenne Connect and a small example of combination therapy looking at that data. So this is for data from Duchenne Connect from a few years ago, and I'm just throwing this slide up to put this into the discussion. And this is looking back at many of our boys when they lost ambulation and what drugs they were on. So were there combinations of agents that were indeed correlated with boys walking longer? So we had data on 384 boys where we knew at what age they are and at what age they lost ambulation, and one could correlate different drug therapies. So prednisone or deflazacort in this data set prolongs ambulation by 3 years. That was the single largest effect size amongst everything that people were reporting into Duchenne Connect. But you could then do multivariate analysis where you take all the data together and condition each data point against every other data point, or each drug against every other drug, and in that process look to see if there's independent contributors. And in this data set, vitamin D administration and coenzyme Q10 administration were statistically significantly associated with longer age at ambulation. And I think some of those data will probably be borne out and are not necessarily so obvious. I'll give you another example, and this is a little bit of a different approach. So this is some work with at our center with Carrie Michelli and Melissa Spencer looking at enhancers of exon skipping. So this is work that we started 10 years ago, and we did it blindly. We said, exon skipping, very interesting. Could we make it better? And just did a blind search through all other drugs to say, what drugs could we find that might make exon skipping better, increase the amount of dystrophin produced? Because we had an assay that was relatively simple, dystrophin induction, you could search across hundreds or thousands of different molecules to find one. One of the molecules that we found was dantrolene, and we're working on that combination. But that's, in many ways, a simple model, right? We fixed one point. We screened across many. We actually screened across many with one drug dose, and that was a lot of work. So can we get to a point where you can do this much, much more efficiently? And to just give you a flavor of the scale of the problem, if we had one drug and we're going to search for 4 different doses, that's 4 different clinical trials. Yeah, that's not so bad. If we had 2 drugs and we're going to search for the combinations of 4 different doses in those 2 drugs, that's 16 different clinical trials for those 2. If we had 10 different active agents and we wanted to do a clinical trial of all combinations of 4 drugs, that's 1 million different clinical trials. Never been done in any disease, regardless of how much and how many patients and how much money you have. So it's physically impossible to necessarily practically explore all of this space, and that's not what we'll do. So can we find ways to more meaningfully search through this space here of a very large number of different possible combinations to say the ones that are most effective for the kids? And I'm just going to leave it there, saying that's where I think a lot of our effort can and should be. So I'd like to open it up for questions and thoughts. So, I don't know, Lee, maybe, or do you want to start?
Yeah, so as you just heard from Stan, the problem is that we can't really combine everything in the kids. So, and right now we can't combine drugs that aren't approved. So what we've begun, which is not a million different combinations, but we've begun to look at combinations where we know the mechanisms are non-overlapping or at least complementary, trying to use animal models, trying to start with mice and look at what's additive, what's not additive, if there's problems combining some drugs and not others. Now, one of the big challenges we're facing is that, in fact, most of the companies, with two exceptions that you heard yesterday, are actually doing a combination trial. They're doing a combination with steroids. And so we have a model that most of us use, which steroids aren't particularly effective. So you can't even combine the drug that the kids are all on with the drugs you want to investigate. So this is one reason we've spent a lot of time trying to find a model that has a lot more inflammation and lacks dystrophin, because we do think it's the sort of blunted inflammatory response that you see in the C57 background that makes that mouse model less than it could be. Now, there are many other physiological reasons why no mouse is ever going to be the perfect model. We have to try to get things that where the sort of the balance of the mechanisms that contribute to the disease progression are as close as we can get to what's going on in the human, so that we see changes that are much more like the human, and then be able to go in and do combinations. And so right now, I told you we have a more inflammatory background. It's actually a hyperinflammatory background that we've got on the dystrophin deficiency. And so we're trying to understand how well that background responds to prednisone. Because if it does respond in a way that's more like the human, then we can start to do combinations not only of new drugs and both approved drugs that are not in common use or not standard of care, such as I won't name any, because anytime I say anything, you guys put it in your kids right away, so I won't name any. But then at the same time, we've got to be able to put these combinations on top of the standard of care, which for the foreseeable future is going to be prednisone. And so the model's a challenge, because as you know, as you heard, you can't do this in kids. We can't afford to do this in dogs. I mean, what we'd like to do is discover the best combinations that the mice suggest, and then put a few of those into dogs and see if it's borne out, and then try to move it to children. Now, the other challenge, of course, if the drug's not approved, we can't put it into a combination. Right now, we can only combine things that are approved, and so we're stuck with that as a limitation once we get to the kids. It's only as these drugs slowly get approved as monotherapies that we'll be able to investigate combining them. And the complications around that are something that maybe you should get Ed Kaye to talk about, because that's a little outside of my wheelhouse.
Perfect. Well, let's jump over to Ed Kaye then. So actually, I will add to Lee's comments that I think in general we move forward with monotherapies, but moving forward with actual combination therapies and getting a combination therapy approved is not impossible. Sort of, Augmentin would be a classic example of this. It's really two different drugs put together that are a very powerful antibiotic. How does that process look from the drug regulatory?
I mean, I think most biotech or pharmaceutical companies would be very resistant to test two compounds simultaneously when you're in the basically where you're in the clinical stage trying to get a drug approved. And the reason is, if you have an adverse event, you're not going to be sure if it's due to your drug or the other drug. And so it's a risk that could really slow down development. I think, but that's why I'm intrigued by Lee's idea of, can we understand early in the preclinical stages what combinations make sense, so then when drugs are available, we can test them. And the challenge we have is, as you mentioned about Augmentin, is one. So it's, what is the dose ratio that you use, because there's no guarantee that the dose that you're using as a monotherapy is going to be the same dose that you're using as a combination therapy. The other challenge that we're going to have to figure out is, we have a lot of interesting therapies. So we have myostatin inhibition, as an example. And if you're using that with a dystrophin-producing drug, when do you start myostatin? Do you wait until you have dystrophin and then you add myostatin on top of it? And the same is true for inflammation. Do you start to dampen inflammation and then add, let's say, a dystrophin-producing drug, or the same thing could be for antifibrosis? So we have to figure out not only the dose, but we have to figure out the sequence and the timing. So it's tough science, but I think it makes perfect sense, because it's not one disease. Yes, it's a dystrophin deficiency, but you have this upregulation of the immune system. You have a fibrotic process going. And so the question is, how do you use this to maximize the benefit? And I think your analogy to leukemia is spot on, because one drug probably isn't going to do it, and it might be 3 or 4 drugs. But I think one of the things we could do is, once we have these products approved, we may have to have something like the pediatric oncology group, where physicians are willing to take the responsibility of testing. But that means you have a protocol, and everybody in the community follows the protocol, because a single site will never have the information that you need. So I think one of the things we could think about is, do we develop a Duchenne study group that people can write a protocol, you approve the protocol, and then you are able to use various combination drugs to think about what makes sense and how does it work. And just so everybody knows, it will take time. This is not easy, but it's certainly a worthwhile goal to think about.
Stan, if I can just want to challenge one aspect of that is, we do combination studies with investigational agents all the time. So the agency will allow combination investigational agents. It's just, as has been pointed out, it's very complex. There's a number of drug-drug interactions you need to worry about, overlapping toxicities, things that you need to consider. I think we're in a good opportunity here that we have a mixture of biologics and small molecules that, typically there are good combinations that we can do. When I liked your example of the HIV area, when we were developing AZT and 3TC, those were investigational agents when they were first combined. And so I think that there is that challenge for us to try to come together as a consortium to try to figure out what those right combinations are, do the models as Leah has pointed out to try to guide us. But I do think that I guess I just want to challenge the assumption that we need to wait until everything's approved.
And just for the industry folks as well. Oh, please, go ahead.
I just wanted to add one thing from the industry side. So the foundation that the FDA is looking for, and what we're all looking for a combination therapy, is that it's better than the individual components. So you need to know what the individual components here do. Drugs for HIV, the drugs that were just recently approved for hepatitis C, you actually had to use those on an investigational basis together because of the emergence of resistance. So that was a different development plan, that you had to do that in the early stages of development. But typically with combination therapy, what FDA and what we're all looking for is, does it work better than the individual agents? Because you don't want to be adding a drug on, exposing kids to additional side effects without knowing that it's better than the individual agents. So the first thing all of the companies need to do is to understand what the efficacy of the drug is by itself. And we're still all, even the drugs that are the most advanced are just at that point where they can say, yes, we understand this, we can go to FDA and ask for an approval based on the demonstration of efficacy. So it's really important that the foundational work from the preclinical is there waiting, so that once multiple drugs become approved, and have established efficacy, then we can ask the next stage of questions, which is, how do they work together? But we have to know what they do individually first.
Before we think about combination therapies, it's extremely important to recognize what we are dealing is not a simple situation. So we often name this particular disease as DMD, and therefore we want to find a single drug for DMD. I don't think DMD in year 4 and year 8 and year 10 are same. It is highly unlikely the processes that are taking place in year 4, year 10 and year 15 are similar. That means we need to have combination therapies that are unique to year 4 and year 10 and year 15. So for that, do we know what combination to use in year 4? We need to invest in finding out what are the processes that are taking place. So this comes to the defining the disease. This is a big problem, not only with DMD, for many other diseases. We need to define the stages of the disease based on the events that are taking place. So you are going to target, the drugs you are going to use are going to effectively target those processes. And I don't think we know this well, but we know this much. Inflammation comes in later stages, you have fibrosis, you have less regeneration, but we don't know truly what are the drug targets. I think it's the time. I'm so glad that we are talking about combination therapies, recognizing that this is not a single disease, and we are dealing with diseases, probably multiple different diseases within the same name, and multiple different processes taking place, and our goal is to target these in an age-specific manner. So in one way, even if you have a combination of 4 drugs, it is highly unlikely that they will work in a kid with 15-year-old if the processes are taking place in a child with when they are at 5-year-olds. So I think it's the time for us to invest in research to find out what are the age-specific targets, and then develop combinations based on that. So that means our thinking need to change. I think it is a misnomer to call DMD. So this work, combination therapies works in HIV because the target there is just the virus. Here, you have multiple different events taking place.
Thanks, Raju. John,
So
I think you want
I think Ed's point in what Raju just said is that timing is everything, okay? So there's that additional variable. I'm going to jump back to the talk that we asked Jill Raphael Fortney to give a couple days ago, I guess it seems now. That the importance of understanding the different mechanisms, the different pathways that contribute to Duchenne, and then understanding which drugs hit which pathway. So I think the model that Lee talked about is something that we've talked about among the Scientific Advisory Committee at PPMD at a meeting just the other night. So Lee had discussed this with Pat and me, and we brought it to the committee, and we're going to do some version of that kind of approach. Because it's so complex, we're going to have to triage these preclinically, but the way it's going to have to work, because it kind of pulls the timing issue out a bit, is finding the best drug for each specific pathway. So like head-to-head, looking at different antifibrotics, head-to-head, looking at different drugs that promote muscle regeneration, and figuring out what hits the target the best and modulates the target the best in the animal. Reduce the complexity, just because there's lots of complexity here. We cannot do these studies we can't start at square one in the clinic for the reason this slide that the slide you had up before about how things multiply out, the number of different combinations. We've got to do them preclinically, but we've got to do them quickly because, particularly as the PPMD representative sitting up here, I want these drugs to be out there tomorrow, right? But we're going to have to go preclinically. But the right way to do that is to ask questions about what are the important pathways that contribute to the disease. And Jill did a terrific job with her talk a couple of days ago. So I hope that came across well. But figure those out. So how are we going to do that? So we're looking at ways within PPMD about leveraging our own funds to try to help support this type of research. Second thing, we've met with the NIH, And my old institute, National Institute of Neurological Disorders and Stroke, does a lot of muscular dystrophy funding. But they also have a preclinical therapy development program that there were lots of grants that have come to this field while I was there. Lee Sweeney had a $15.5 million grant for therapy development that came out of that specific program. The problem is it's more geared toward monotherapies. So Justin Fallon, Justin's still here. His grant is right there. His grant is developing biglycan. He got it through that program. But that program is really geared toward monotherapies. So we knew that, and when we met with NINDS, we said, how about considering a combination therapy initiative? They said they're certainly interested in it. We're going to keep pressing them to do that. The other thing we want to do with the NIH is try to do exactly what Stan is talking about here. Try to bring in expertise from fields that have already done this instead of trying to reinvent the wheel. From oncology, from AIDS, they need to help inform the rare diseases about how best to do this, and we're taking that approach. So we're really open to ideas. We would love to hear from every company at this table and out there in the audience. But PPMD is really committed to trying to push this approach as strongly as possible, and we're trying to leverage resources with the NIH and other places as well.
Thanks. And then, Craig, I think
I think we have to realize that really combination therapies is really here. The dystrophin restoration strategies are all being evaluated in the context of underlying steroid treatment. That's an inclusion criteria for those trials. We don't exclude patients who are on afterload reduction with ARBs or ACE inhibitors, and those have known skeletal muscle effects. So I think it makes really no sense. Monotherapy doesn't make a lot of sense to treat a disease as complex as Duchenne that has skeletal muscle dystrophic changes as well as cardiomyopathy. And I think that approaches that really look at the pathways in terms of fiber integrity and structure, optimizing metabolism. You've heard a lot about targeting the inflammatory component through the NF-kappa B pathway with less toxic agents. And then there's going to be a need for other approaches looking at fibrosis and potential for muscle growth as well. So I think that combination therapies will be the future with Duchenne dystrophy. They already are here in that we are studying patients on multiple agents. I think the animal models that Lee and Raju and others are involved with are critical in sorting these issues out. I think the biomarkers and the validation of biomarkers that can be used to actually assess treatment efficacy with plasma biomarkers, imaging biomarkers, ability to actually assess dystrophin levels and utrophin levels are all going to be really quite critical. But I think really combination therapies is a critical issue, one that's here and one that we just need to address head on. But certainly we can be informed by the preclinical studies with the animals and high throughput screens and so forth.
And actually, I have a question for some of the industry folks, but also the scientists, in that is there a role for repurposing drugs that have not made it through? We hear about the 1 in 12 drugs making it through, even once they've been in humans, so they've made it a long way down. And if indeed some of the studies of the combinations are not necessarily optimal for taking the best dose of drug 1 in a given category, taking the best dose of drug 2 in a different category, combining those is not necessarily the route by which leukemia has been most effectively treated. It's a meandering route, and it's a 50-year route. And it really is a clinical trial route, but combined with some data supporting each area. Is there a meaningful route for exploring that data space in terms of preclinical models or even cell line models, but then with the idea of bringing actual very complex combination therapies, or is that just too difficult to place? If we don't move forward into clinical trials as monotherapies, we can't possibly move forward with combination therapies as a strategy.
Just to make a comment on that, in fact, this is done all the time. And a lot of times there is a compound that goes, let's say it's used for cancer. It's not found to be very effective, but it has a particular target. And someone says, well, I need a drug for that target. And so then they'll buy that asset from another company. It has a lot of preclinical toxicology. It might have human toxicology. And so it is a way to accelerate it. So I think it gets back to your major focus is that we really need to understand the targets. And then you can actually go into, there's a lot of drugs that are targeted for specific purposes and maybe in a particular disease that are worth bringing off the shelf. And sometimes they're on the shelf for 10 or 15 years, and someone has an idea that maybe this could work. Let's test it in a different disease. So that does happen all the time, and I think it's a matter of just knowing what are the compounds. And sometimes the only challenge is, of course, each company has their own compounds, and it's not like it's public knowledge necessarily. But if you know someone has an asset that could be useful in your disease, that is many times a faster path to get it into the clinic because there's already a lot of work that's been done. So, yeah, I think it's a very good idea. But that gets down to the basic research. What's your target? What do you want the drug to do? And then you have to test a number of different compounds to see which one's the best.
I'll disagree. I fully agree with you. It's there's examples out there. So, there's drugs like 3TC that were dead because the resistance developed so fast to them. But we found that in the lab and also in the studies that combination with another drug, AZT, you had efficacy. And it turned out to be very, one of the most critical developments in HIV was the Combivir. So it was one drug rescuing another that enabled it to come in. So I fully agree, and there's examples out there.
One of the things that actually I think is probably useful to do this morning, and unless people have specific other comments that they wanted to say, okay, sorry. So I think I'd invite people to come up now to ask questions because I think it is a complex area. It's one of the questions that you'll be having for your own children. There'll be very simple examples of idebenone may well get approved for Duchenne this year. And if your child's on coenzyme Q10, would it make sense to have somebody on both of those agents? Some of the logic that you've been hearing, the answer would be no, because they're effectively doing the same thing, in which case you'd choose one or the other. And so I think some of these things will be playing out in terms of if we know the exact target that a given drug or category of different drugs are doing, you'll probably not be using a combination of those. However, I would say you could argue that both of the agents that were just being discussed as antivirals have a very specific target, and yet they still needed to have two different agents. So I think in some ways we'll unfortunately still be learning a little bit by trial and error. But Greg?
Yeah, I think it's important to consider that even though we may be talking about one target, I think there may be different small molecules that safely could hit that target. If you're looking at idebenone, for instance, it's targeting the mitochondria, it's targeting reactive oxygen species and calcium influx that occurs that actually CoQ10 or idebenone could potentially target that, but perhaps another approach that induces mitochondrial biogenesis, for instance, epicatechin, which you'll hear more about later today, may be another mitochondrial target, but may actually make rationale to actually use both those agents to target mitochondrial health and improve oxidative health and metabolism of the fiber. So I think there could be multiple agents that could still safely target one pathway and be effective.
Stan, the other example of that would be drugs that hit different places in the same pathway. So, like, there are drugs being explored for utrophin upregulation that hit different points, may affect message, may affect protein stability. Mm-hmm. If it hits different points, then they may be able to be synergistic. Okay, so I wouldn't just say that we need to get the best drug for each specific pathway, but I'd agree with what Craig just said, is that there are synergistic possibilities within the same pathway.
Very good. Thanks. So, I would invite people to come up to the microphones and ask questions now, please. And then I think that'll provoke more discussion as we go along.
Good morning.
I can't see who that is.
Mike, more comment than question, but I'd be interested in the panel's opinion on standardization of therapy. One of the most widely prescribed drugs for Duchenne muscular dystrophy are glucocorticoids, and we all know that there's a whole host of negative side effects that come along with this. And many families are seeking for alternatives, but yet when you're talking combination therapies, you, in my opinion, and from my understanding, is you build on what you already need to know, or what you're already doing. And my question is are there any thoughts about building on prednisone? Because it seems to me that some of these therapies may be synergistic or may be additive to what prednisone is already doing. Excluding prednisone, if the landscape changes, I mean, any new drug that comes, there will be side effects as well, and those will have to always be taken into consideration. And what recommendations would you have for the community about ensuring some sort of a standardization of practice so that you actually can read what is being accomplished?
I'll start with an answer to that, and I'll use actually the childhood leukemia reference just because I'm most familiar with it, but I'm sure there's many in medicine. And the way the field has evolved over time is very much along the lines of what Ed was referring to, is having very large international, multinational, sometimes there's multiple different international consortia, choosing what is the best, most logical experiment to be doing. And it really is a standard therapy compared to another therapy, and oftentimes the protocols would be 4 different therapies varying 1 point. So what I think will happen in our world is we're already in, exactly as Craig had said, a combination therapy era. We're already doing it for our children. Your physicians are already doing it. We're doing it with agents that are not necessarily as effective as we'd like, and we hope for much more effective, much more thoughtful therapies coming along the way, better mouse data, better cell culture model data supporting them, and actually better monotherapy data supporting them. And then the question is, how are you combining those? It will unquestionably be on top of what's already happening. I can't imagine a physician suggesting at this instant, because you're going on to exon skipping, I'm going to take you off of steroids. If there is, happy to chat. Happy to get Craig's perspective there. But I think it absolutely will beg. It would be such a complex space that we would be better off with a set of very large studies where every child is coming in onto one of a series of different regimens so that we iteratively learn what is optimal in a very rigorous way. Raju's points are that could look different if we're enrolling 1-year-olds, 2-year-olds, 10-year-olds, 20-year-olds, because the disease is changing over time. So it's not going to be a simple landscape as it is in cancer, actually.
Well, I think we know that there are boys and men with Duchenne that are being treated with less than optimal steroid regimens. But we see these patients all the time that come to us. I think the concept of the pediatric oncology group, where you have known protocols that are adhered to and can then be objectively followed over time, I think make complete sense. And in a sense, when you look at the 4DMD trial, which is really objectively evaluating 3 steroid regimens as to efficacy and toxicity and safety, I think that in many ways is almost the first model that we want to proceed with. I think over time, those patients are all being treated, but with regimens that have scientific rationale. The investigators are adhering to a known protocol, and I think that's a nice model moving forward. And if we eventually develop a Duchenne study group or something analogous to the pediatric oncology group, I think those are the protocols that are eventually going to be developed and are going to be adhered to by the treating physicians and centers.
Yeah, I'll add a slightly different attitude to that. Is one of the benefits in pediatric oncology is, from my perspective, when I used to treat kids in that space, was almost every child was on a specific clinical protocol. It begged standard of care. It begged specific follow-up and maintenance and observations that we as physicians, when we're just treating a child in our clinic, have great latitude for how we do it. We love latitude. Patients love latitude. Clinical trials are harder, but they allow us to gather the data in a systematic fashion, and they force our hands to gather data in a systematic fashion. So oftentimes when I was consenting a patient where we had a gold standard, very effective therapy, and we're going to possibly randomize your child, because I don't know what the best thing is, onto 4 different regimens, you got to be kidding me. I'm going to go take a gamble with my child, and you're telling me there's a therapy that's 80% successful, and maybe you can do a little bit better with 3 other options? It's not so compelling as a parent to say, oh, I'm in. But what I think is compelling is I believe, and it's very easy to tell parents, that being on the clinical trial will probably get you better care, because you're not just going to be getting the gold standard. You're not going to be just getting standard of care. You're going to be getting an observation across a study that was designed by the 20 best leukemia doctors on the planet. And worker bees like me in the clinic at the time were going to be using that information in a very systematic way, and we would abide by the study protocol. I'd argue we've already seen that in the 4DMD study. I'm not sure who was saying it yesterday. It's vanilla. It's not a very exciting study, but it is a study, and I'd say the people that are participating in that and the clinics that have already seen a change in the behavior of the physicians and the treating teams because of those kids are on this trial. That's a good observation.
Yeah.
Thank
Stan,
you.
I think the 4DMD is a good example, because we have mandated that boys be on steroids for many of our trials, but we didn't say what type of dose, and the reason is I can't tell what dose is best. So actually having a study that tells someone, is prednisone not as good as deflazacort? Do you have to do it every day as opposed to every other day or weekends only? And that's important information. And then, again, it standardizes the dose, and then you can add an additional therapy on top, and then you know what your drug is doing. And I think one of the things that's going to be interesting, as drugs become available, one of the things that companies can do is, you have an arm that has to be on a Debenone or whatever, or isn't on a Debenone, and then you can compare the difference. Is it synergistic? It's challenging, but it's one of the things that we can try to control. And one of the things we're doing in our study, looking at younger boys, 4, 5, and 6, they can either be on steroids or not be on steroids, because that's not everyone at 4 years of age is on steroids. So it'll be a way for us to at least get an idea, is it synergistic with exon skipping? So we have to get that information, and the more regimented we are, then the better we can understand is what is the effect of your drug, because you're only changing one thing at a time, not 2 things.
Please.
Hi, Mike Boss. One of the complexities that we're having to deal with in DMD that's not been discussed as yet is the variability of the endpoint in comparison to a viral disease where you can just measure viral load. And we've got such variable and difficult to measure endpoints, it makes combination trials particularly tough.
No, I'll agree with that. Something I was been considering is the standardization endpoints. We're having a hard enough time standardizing it in monotherapy, but understanding the mechanism, and this is a multi-organ, multi-pathway disease, that it is going to be much more complicated than tying a bunch of immune modulators together just to hit that one target. So it will be something that we will have to strongly consider co-primary endpoints and work with health authorities to think about how we look at these different and standardize.
Yeah. And I think Lee had also mentioned it earlier, having endpoints that might be observed earlier in the disease process, having those be robust. For instance, in the dystrophin restoration strategies, having dystrophin itself as a very effective biomarker would be critically important for moving things forward, and having the regulatory process accept those easily is also an important aspect, which I'm sure guides industry's hands. Please, I think you were.
Thank you. What I'm hearing from you is that we may be as much as a full generation away from knowing what the true combination or what the right cocktail is going to be for our kids. What advice do you have for parents today Some of these drugs I'm looking at and listening to, these are already approved, maybe not for Duchenne, but they're available. And we don't all have the ability to go to a Cincinnati or a Columbus or a certified Duchenne care center. And so we become the experts in our children's care, and we're the ones going to our doctors saying, hey, I heard about this drug. What about tadalafil, or what about idebenone? Is there any advice for us today in how we can start utilizing some of these drugs that are available for us, and things to look out for, things to avoid? If we put our sons on idebenone, is that going to stop us from being eligible for other clinical trials if they become available? And anything that we can do today.
Craig, do you want to start?
Well, I think optimally, I think discussions with your treating physicians in terms of the evidence-based approach to these treatments, I think makes complete sense. I think that if you're looking at a drug such as tadalafil, for instance, which we really don't know the appropriate dose of tadalafil. So in my mind, a medication like that, and that medication does have potential for some side effects. I mean, I think that drug really needs to be studied in the context of the clinical trial, and we'll be obtaining that information shortly as to the effectiveness of the therapy. There's 2 doses being tested in that therapy, and if we see efficacy, then there'll be an approval that hopefully will be soon to follow. Then the question is going to be, if you have a child that perhaps doesn't meet the entry criteria for that trial, a child who's non-ambulatory, for instance, would tadalafil make sense at that point in time? So I think those questions are going to have to be certainly addressed. With regard to some of these off-label medications, there is oftentimes the opportunity for adequate washout to allow participation in a future clinical trial, and that's always a possibility.
Yeah, and I think it is hard to give specific guidance, partly because it is a complex world, in terms of the number of different options that are theoretically out there. And I think the route forward is not everybody has to be on a clinical trial for everybody to benefit from the fact that clinical trials are moving forward. So, we will learn about these agents, even though we might study a reasonably small group, even though the disease process is changing over time. Would I extrapolate as a treating physician very readily to say what's best for an individual patient? Absolutely. So I think, even though the disease process might be different at 2, some of the treatment strategies we will absolutely want to push to birth for when those treatments will, the logic will be very clear. Others we won't want to start until particular parts of the disease process. And it is, I'd say, incumbent on us to make it so that there is a process where everybody can make it to a site which can participate in these large trials. I think that'll be absolutely on the community to develop. So I'd say that's inadequate at the moment.
Yeah. And one of the concerns, I think, and there's precedent in other diseases. One disease area that is prone to multiple medications is mitochondrial diseases. And people over the years have thrown absolutely everything. And I recall, seeing many of these patients. They will come in on 15 different drugs, multivitamins, coenzyme Q10, vitamin E, high doses. And if there's something that's working, you would never know, because there's no dose regimentation, there's a number of drugs, and also it could be potentially causing side effects. So it's just a slippery slope that, you say, well, we've got to do something, and I have to throw something at them, and I have to do it, I have to make them better, but you may be making them worse, or you may have a drug among 15 that's actually working, but you'll never tell. You can't sort it out. So it's one of the things we have to be, I think it is a difference in philosophy. Are you more scientifically rigid, or do you throw the kitchen sink? Sometimes you get lucky when you throw the kitchen sink, but many times you don't, and you could do harm.
Yeah, I'm sorry, probably not completely satisfying answers for that. Please.
While it doesn't seem to fit the definition of a combination therapy, how would the FDA interpret multiple exon skipping? would they view it as combination since it seems to have multiple aspects?
Yeah, I think that's in your court.
Yeah, I believe they would, because again, it's two different drugs, but I think those type of studies could be done because if you have an idea of the dose for each one, then you could look at that, or you could look at variable doses and actually study that. So it would be considered combination therapy. One of the interesting things that Steve Wilton, who many people in this room know quite well, but some of the animal work that he's done with combination drugs suggests that you in fact need to lower the dose when you do dual exon skipping. So that's again one of the reasons why it would have to be studied because you can't assume that the dose is going to be the same when you give it in combination.
There's more of a question of timeline. That's what I wanted to know.
Okay. Yeah, so I think it would my impression, again, we haven't had that discussion with the FDA, but I think it would be, you'd have to test each one individually, and then once you know what the effect is, then you do the combination studies.
Well, first, thank you so much for your valuable time, hard work, and insights. We really appreciate it as parents, so thank you so much. My question is around creatine monohydrate, and physicians have differing opinions about the use of creatine. One physician we met said, fine, I do advise my patient, it's over the counter. Our physician who we are working with suggested that it may be an exclusion criteria in the future, we don't know. This is a specific question for Ed, and because we are waiting for the Sarepta trial, the 45 trial to come, and we've avoided giving our son that, so we don't know what your stance would be on creatine monohydrate.
Yeah, I think the short answer is we really don't understand what the effect of creatine would be in the muscle, and what would the dose be. Again, it's been used for performance enhancement in athletes, and the data it's unclear what the effect is. I think, but for something like this, which would have a fairly short washout period, if it was done as part of a clinical trial, there would probably be a washout period that would be necessary to get rid of it, so you don't have that as an interfering drug when you're doing a clinical trial. So it wouldn't be something that would really interfere someone from entering a trial, as long as we'd have to know what the inclusion-exclusion criteria would be and how long you'd have to wash out that drug. And usually it's a matter of a few months, and then that would be fine.
I see. Just a follow-up question on these supplements. Is there any trial which essentially suggests a combination of supplements rather than instead of drugs? I mean, drugs is a separate thing, but supplements are available over the counter. Any study around what combination works better?
I don't know whether not to my knowledge in terms of a very large systematic sorts of studies. I think that it does beg a very good question to do that. I think there's many people. There's evidence that some of these nutraceuticals or supplements indeed may have some efficacy, and indeed because they're accessible, many people will use them, and we have a relative paucity of information. One of the things that was certainly discussed amongst PPMD, because it's an often parent sort of stimulated question exactly as you're doing, is what do we really know? What do we know about these bits of information? And the literature is actually quite vast and quite unclear, I would say, at the end of the day, which makes it very difficult to wade through in a really clear, crisp way and come up with sort of physician recommendations for exactly what would be best, or even what would be the best study to do. But I like, this is my opinion, I like having data like Duchenne Connect data. I think some of these things are back in our hands as a community. If there were, let's go for the optimistic side, 50, 60,000 people who had reported their data in and accurately recorded how much of different agents they were using, would scientists look back through that? That would be an interesting data set to look at. There'd be papers published on it, looking at the interactions between these different supplements. Would be possible to do to stimulate the search through that space that's cost effective and to provoke specific clinical trials that could be done in a very focused way. Alternatively, you're looking at really complex, expensive clinical trials that I don't believe will be easy to fund. I don't know if others want to chime in on that. Ed, you've been in this space.
To me, one of the interesting things when we talk about supplements, because it's so variable, that's where large data sets might be able to help you. So, we were talking last night about critical path. If we had a large number of patients and we actually knew all the supplements, you might be able to pick out a signal. So that may be a way to get, some scientific basis or why you would give a supplement and be able to tease that out. So again, I think that is another point to try to really push, combining data sets, looking at critical path, and what can we learn when we have large numbers of patients as opposed to small studies.
All right. Final question, I promise. Is it fair to presume that supplements that don't directly affect the muscle function is all right to use?
In regards to being in, for instance, a clinical trial like...
For a clinical trial. And the reason I ask is, my native country, India, has an entire medicine system of Ayurveda in place. And a lot of that has to do with cleansing of the body, and because of the usage of such high dosages of steroids for a child, it helps cleanse their system, if you will, and reduce the acidity that's caused and all of that. It does not impact the muscle. But having grown up in that environment, we know the benefits of using a digestive supplement, if you will. But we've been completely avoiding that because as a parent, you have this dilemma where you know that it can benefit your child, but we are so afraid of using it, saying, don't know. We don't want to be excluded from a trial, so we're just avoiding all of these things, even though we know the benefit. Our doctor's perspective is, as long as it does not affect the muscle, it's not directly impacting the muscle, it could be okay, but they tend to refer back to the drug company saying, don't know what their inclusion or exclusion criteria would be. So that kind of keeps us in a dilemma. We want to help our son, but we don't know what to do. So is it fair to presume, as long as it does not impact the muscle directly or indirectly, is it okay or not?
I would say maybe, but Joanne, do you want you're doing a clinical trial right now. How would you feel?
I think that's hard to answer for all supplements. I think what we do know from the Duchenne Connect database is we're not seeing a strong signal for many of these supplements. And that's the beauty of a randomized placebo-controlled or a randomized trial, is that you are then averaging out many of these small changes in each of the groups. So, for example, we have not told people to stop taking supplements because none of those supplements have shown a strong signal to date. So that's been our philosophy. I think other companies may see that differently.
I think the only thing you want to watch out for is there anything that would impact the absorption of the drug, metabolism? Those are things that's more of a concern, is would it impact or send a safety signal? And the problem is the lack of data that would to support that.
Sure. And in general, would you be okay if somebody was on probiotics, say, and they stayed on the same probiotic regimen? Would that be a no-go for an oral drug, or would you just say, good enough, keep going, just don't change it?
Yeah, I mean, I guess it depends to say if it impacts metabolism or absorption. I mean, that's where you'd really want to understand that. So we would do a profile to understand what the potential risk is associated with that.
Very good.
Thank you so much.
So I guess I just have a concern, back to the mom's point about what can we do today, because I look at the list and I do think that theoretically we do have kids that are going to be coming off their first bout of the trial in Adalurin or eteplirsen or drisapersen, and some of those kids are on a eplerenone, and I'm worried that we're not capturing the data. And Stan had said that, if Katina is approved this year, we'll have kids on 3 of these drugs, and we're not really capturing that in a systematic way. I guess that's my sort of concern. And my other concern is that all the trials seem to be focusing on the ambulatory kids, but yet we've got this data set of the now non-ambulatory kids who are also going to be the examples of the combination therapies, but we're not really gathering their data, so it's going to be usable. I guess those are my concerns.
Yeah, I think we all share those concerns, absolutely. And I think the flow forward is looking for strategies that allow us to answer those questions in a much faster way. Some of the issues, again, sort of drawing out the childhood leukemia strategy, was it was all done with very large, very slow clinical trials. It's not necessarily the model that we want to follow completely here, and we'll learn different things from the AIDS world, we'll learn different things from the cancer world. They're not the same disease processes, but each of the main clinical trials is it's an 8 to 9 year process per regimen. So I don't think that's the world we want to be in here. And I think there's strategies that we can probably explore this space much more quickly. But yeah, I think of it as it's a step forward where at least there's multiple effective therapies where we even have this question that we're faced with, and that's fantastic. So.
Yeah. Just to echo that point, and the reason that those advances were made was because of high double-digit participation, unprecedented in any other disease. And though I see that this community has that potential, and we see that interest, we know that these trials are a burden on parents, on boys and men. And we try to help as much as we can, but the burden rests on you. It is so important to have high participation to advance all of these drugs forward, and so we thank you for that.
Yeah, and I'll echo that as well. Is that I would say the way we as a community can probably make progress better is not only allowing, but really pushing for clinical trials participations. If there's an add-on study that requires an extra blood draw, my answer is always yes. Dylan always agrees. And I think we want to make this environment as permissive as possible to make progress occur as fast as possible. Leukemia doesn't have that same luxury in the sense that everybody's put on a protocol whenever it is that they happen to be diagnosed, and you're at the mercy, for better and for worse, of exactly what that rate is for how those kids are coming in. Here, we'll be pushing the issue in terms of the kids are generally already diagnosed, and it's our decision whether we're deciding to put them on a clinical trial or a different clinical trial. And I think doing that a bit more systematically would actually be very good for us.
Hi, I'm Diana Escolar, and I'm from Kennedy Krieger and Akashi Therapeutics. We have several hats. So I just wanted to make a couple of comments. One is that we need to learn, this is Duchenne, but it's of course not the only disease. There are many other diseases. And as Ed was saying about the mitochondrial world, we can learn from other diseases. And in my former life, I did ALS research, Lou Gehrig's disease. And something that we learn very early on is that when you participate in registries, we can learn a lot of things without having to do clinical trials. So we're not going to be able to do clinical trials for each one of these supplements. It's just not enough resources, money, patience, and time to do that. But when the ALS registry started, these patients were taking 15, 20 supplements, and it was very easy to look at the individual sites, what they were using, and discover very simply that all of those supplements were really not working in ALS, and the care really changed. So please, please, please participate in the Duchenne Connect registry, and not just entering your data once, but every year as they ask you to, because we spend here year after year trying to collect data, and the more we have the data, the more we're going to know the answers of these questions. So I just wanted to emphasize that.
That's great.
The other one was about combination of supplements. So the only study that I know that did that was an MDX mouse, and that happened. Grant Shelley did that in Buffalo, and that was maybe the trigger for the Synergy Network. That's how we started that. trying to see if any of those 40 supplements and drugs that he mixed and matched and used individually were working or not. And I can tell you that some of the supplements that look like nothing, they actually made the mouse worse and not better. So it is important to, have some idea about what you're using and consult a little bit more, not put everything on the, kitchen sink. And my last point is for the industry folks in the in the panel probably, and somewhere else. I think that more important that washing out supplements is important to have a stable baseline on the patients before they come into the trial. And some of the supplements are disease-modifying. We did a study of creatine and glutamine. It was a randomized placebo-controlled study published 2005 in Annals of Neurology. It did show that creatine modified disease progression. The Canadians also show that. Not statistically significant, not a huge effect, a tiny effect, but it was a short trial. So we don't know over the long term what it's doing. And if you just wash that out 3 months before you start your trial, you don't know what you're doing. I mean, maybe you are changing the baseline in that kid that has been for that for 4 years. So I think that just asking for a stable baseline of everything that they're taking and they don't move anything, don't change the dose, and that definitely includes the corticosteroids, for at least a year before entering the study. That's more important than washing out.
That's great. Thank you, Diana.
Sri Pandya, University of Rochester. The only thing I wanted to say is, just like you have the Duchenne Connect registry, I want to make sure that all of you are also aware of MD StarNet. I'm not sure if Julie Boland or Natalie Street are in the audience here, but as a co-PI from New York of MD StarNet, you know that it has over 1,000 boys with Duchenne dystrophy, and we have data from 1982 onwards on these kids and young men, and that it's abstracted data from medical records. So it's a huge resource that I hope we can all use.
Yeah.
Thank you.
Thank you, Sri. That's fantastic. Thank you very much. Let's give unless there's final comments? Nope? Okay. Let's give our panelists a round of applause. Thank you guys.